Alright, in this video I'll explain to you how to look at some protein characterization and some parameters for protein identification and modification. So again, we've got this sequence here that doesn't look like anything, so we need to first, this is a nucleotide sequence, so you're going to first use this sequence, and you're going to copy it, and we're going to translate it into a protein sequence so that you can use other programs for more information. This is the sequence is the virulence factor of Staphylococcus aureus, but we're going to first use our BLAST-X so that we can get the protein sequence from our nucleotide. Okay, just passing the nucleotide sequence, say BLAST. Nope, I have to delete that information here. Let's see if it works now. You have to be really stubborn doing bioinformatics. Alright, so we took the nucleotide sequence of our virulence factor and we are translating it into a protein. And there's a couple of different ways. One is the BLAST-X, but the next tool I'm going to show you is an ExPASy tool, which is a Swiss bioinformatic database, and it's also fantastic, lots of free, easy to use tools. Alright, let's give it a second here. Alright, while we're waiting for the NCBI to start, I'm going to also paste it into the ExPASy tool. Let me make sure you see it. That's the ExPASy tool here. I pasted the sequence in and I say translate. Translate tool. You put the sequence in, you can use the translate, and then you scroll down and again you find your different reading frames. And as in a previous video, you need to understand the 6 different reading frames. It's because we don't know exactly what nucleotide the translation starts, and there are 3 nucleotides per amino acids. So each of the start possibilities makes 1 reading frame. The other 3 reading frames are coming because it can be read forward or backwards. So the DNA doesn't have to be encoded just reading forward, but it can also be encoded reading backwards. But when you see the results, it's pretty obvious. So to scroll through, you have the 6 reading frames, and there's only one that is read that basically is a whole protein translation. The other ones are just very short start and stop codons, so you can ignore those. So the frame 1 would be the one which would be your protein. Let's go back to our NCBI BLAST, and it's gonna be probably the same thing. It's gonna tell you that it's a virulence factor for Staphylococcus aureus, and it should be the same sequence as this one as well. Alright, so we're gonna have our sequences translated, and now we're gonna use to analyze them. Alright, so we have now this sequence of the protein through the 2 different programs, the protein translation tool in NCBI. So we can copy this one, or we can copy the one from ExPASy, either one. This should be similar. And now we're looking at ProtParam. That's Protein Parameter Database. And let's computate the parameters, and it will give you all sorts of different information about your protein. It gives you the number of amino acids, the molecular weight, theoretical isoelectric point, so that's really important when you purify the protein, number of amino acids, charged residues and so on, atomic composition, and also extinction coefficient for spectrophotometric analysis, and half-life. So it's really important parameters when you're actually working with the protein. So again, just a very easy copy and paste information for easy analysis of your protein by a couple of clicks of information. Another little tidbit you may be interested in looking at protein structure is not just analyzing the protein information, but also looking at the three-dimensional structure. So if you go to the NCBI, the structure database, and say Mercer resistance, or put in the sequence of the protein you've just analyzed, you can actually look at the three-dimensional structure of the protein, which is really pretty cool. So full feature of the 3D viewer, and you can look at the resistance protein. So crystal structure of PPB2A from Mercer in a complex. Here we go. All right. So tons and tons and tons of information just with the click of your mouse, and you're all becoming better and better bioinformaticians. We do have a minor in the department in bioinformatics that teaches you a lot more about these tools, and they are important for all different applications as you've seen in our introductory video. All right, this is so much for protein analysis. A lot more, just a tiny little scratch on the surface. See you soon.